Good evening, doctors. On behalf of Acumentis Harmonica, myself Shubhojit Mukherjee, welcoming you all. I take the pleasure of introducing today Dr. Jaideep Malhotra. Dr. Jaideep Malhotra is our course coordinator and moderator FIGO. Dr. Jaideep Malhotra is the director ART Rainbow IVF. She is the president Indian Society of Prenatal Diagnosis and Therapy. Dr. Jaideep is the president of South Asian Federation of Menopause Societies. Dr. Jaideep is the immediate past president, Indian Society of Assisted Reproduction. She is the past president, FOXY, Aspire, IMS. Regional Director, South Asia, Ian Donald School of USG. She is the professor, Dubrovnik International University, Croatia. She is the editor-in-chief of SAFOG and SAFOM's journals and many books. Dr. Jaideep is the past vice chairman, ICOG. She is the member of FIGO Committee of Reproductive Endocrinology and Infertility and FIGO Working Group on RDH. Dr. Jaideep is the recipient of honorary FRCOG and FRCPI. Dr. Jaideep is the recipient of Nepal Samman Woman of the Year Ethicon Fellowship, Indumati Javeri Prize, Dr. M.D. Hansotia Award, Best Committee Korean Award. She is the recipient of Jagdishwari Mishra Award 3 times. She is the Forbes Most Successful Woman and winner of Prime Minister Atmanirbhar App Challenge. Dr. Jaideep is the initiator of Club 35 Plus, Adbhut Matritva, and IMOS app. She is credited with first 500 IVF babies of Nepal, first IVF ICSI TESA twins of UP, and she is currently the advisor to Maulana Azad Medical College, JNMC Aligarh, and SMS Medical College, Jaipur. Over to Dr. Jaideep.
Thank you, Shubhajit. Very good afternoon, morning, evening to everyone who's there with us today. I'm Dr. Jaideep Malhotra from Agra, a member of the Reproductive Medicine and Endocrinology and Infertility Committee of FIGO. And welcome to all of you to this wonderful masterclass on endometriosis. Thanks to the FIGO REI committee and thanks to our wonderful faculty from all over the world, and along with that to Acumentis. Friends, this has been a very unique opportunity for us to have this masterclass, which has 6 modules of 6 lectures. This is the second lecture today. We've already had the first lecture on endocrine and environmental disruptors affecting women's reproductive health, with emphasis on endometriosis by none other than the most wonderful speaker, Linda Judice, on the 7th of June last month. Now, today it is going to be a challenge in understanding the pathophysiology of endometriosis, and we have a wonderful speaker from Australia, my very dear friend, Mary Louise Hull, who has tremendous contributions in the field of endometriosis, which you're going to hear in a short while from now. And then we'll have the third module on dilemmas in diagnosis and management by Dr. David Adamson from US. And the fourth module will be on re-evaluating the response and failure of medical management of endometriosis, which will happen in the first week of September by our Irish member of this committee, Edgar Mucano. And then we will be discussing ovarian stimulation protocols in endometriosis and ART in October first week, which will be dealt by me. And the last on the list will be controversies in deep endometriosis by Rui Ferrian. So this is what our whole masterclass is all about. And at the end of all these 6 lectures and modules, the attendees are going to receive a certification from REI FIGO, and I'm sure all of you are going to look forward to this, because these modules are really going to throw a lot of insight into management of endometriosis. We have our course coordinator, none other than the chair of the FIGO REI committee, Yvonne Des Yamal. Yvonne is a wonderful, wonderful friend. And can you imagine, friends, this is 4 o'clock in the morning in Colombia, where she is from, and she has joined us from there. She is a gynecologist and obstetrician, a specialist in human reproduction and gynecological endocrinology. She is also professor at the Newark Grenada Military Hospital and University and head of the Department of Obstetrics, Gynecology and Human Reproduction, also at the Hospital Militaire Central in Bogota, Colombia. She is the scientific director of the Medical Assisted Procreation Fertility Unit of the Mali Clinic and also the past president of Colombian Federation of Obstetrics and Gynecology. Yvonne, the stage is all yours. I'm looking forward to you sharing your thoughts about REI committee and this masterclass.
Thank you, Yadib, and thank you for everyone be here for FIGO and our committee, Reproductive Medicine, Endocrinology, and Infertility. Is very, very important. The education around the world, and of course, endometriosis is very, very important pathology in reproductive medicine and the pelvic pain. Thank you very much for all be here and for the second session in the masterclass endometriosis. Welcome. Thank you, Jaideep.
Thank you, Yvonne, for being there. And please stay by because we are going to have some fantastic discussions after this lecture, wonderful lecture. It is my proud privilege right now to introduce our two chairpersons for this particular lecture, which are again two great people from our country, Professor Pratap Kumar, a brother, a dear friend who's been the head of the department and of reproductive medicine and surgery at Kasturba Medical College, Manipal. He's also been the national vice president of the Federation of OBGYN, or FOXY, what we call as, in the year 1999. He's inspector, examiner, and advisor at the various national board examinations, and also an examiner for undergraduate and postgraduate in several universities in the country and all over. He's also been appointed on the editorial committees of several journals. Pratap is a true academician, as I have known him over the years. He has contributed to a number of publications. He has more than 300 publications to his credit and has presented more than 800 presentations in national and international congresses, and has been the editor of 10 textbooks and contributed several chapters. Pratap, it's wonderful to have you here. Our second chairperson is Professor Naren Malhotra, and I'm sure everyone also knows him. He's also been the past president of FOXY and past president of ISPAT, ISAR, and right now is currently the vice president of SAFOG and member of the FIGO Guidelines Committee, and is a professor in the Dubrovnik International University and also the director of Iron School of Ultrasound. Naren has also published and edited many books, almost about 40 books. He's been the editor and has also been the editor of the SAFOG journal in the past. And he's a practicing obstetrician gynecologist with special interest in high-risk ultrasound, laparoscopy, and ART and genetics. So the stage is all set for us to have our speaker today, none other than Professor Louis Hull from Australia. Louis has been a very special friend, a very, very dear friend, and we have been associated right from Aspire days when I was the Aspire president, and I have seen her very diligently working in the field of endometriosis. And as soon as this masterclass was taught, Louis was there in my thoughts because I know that her work has been very special. She currently is a fertility subspecialist at the Embrace Fertility Associate Professor, leads the endometriosis group and also is there in the Robinson Research Institute, acting head of the Department in Discipline of Obstetrics and Gynecology. She is a consultant staff specialist in gynecology at Women's and Children's Hospital in the University of Adelaide. Louis, the stage is all yours. We are very eagerly waiting to listen to you making the challenge of understanding the pathogenesis of endometriosis easy for us. Thank you very much for being there.
Thank you so much for inviting me. I'm just going to get my PowerPoint up, and hopefully this all works. And I'll just come back to the beginning. So the topic I've been asked to speak to tonight is on the challenges in understanding the pathophysiology of endometriosis. And there are a lot of challenges. I've just put my conflicts of interest there, but the main problem with endometriosis is that surgery is such a critical part of classifying women with and without endometriosis. And that has really put a lot of barriers in terms of being able to classify who should be in a control group and who should be in a group of people with the disease. And that's been quite a challenge to overcome. We've needed to have animal models because you need surgery to look at lesions. It's impossible in humans to examine them over time. So if you want to look at lesion development and disease progression, you need to look at models. And the models are really interesting, and there are a lot of different things you can do with them, but they don't represent human disease. And the last thing is that we have found many, many differences and factors that are associated with endometriosis. And one of the things that we really need to think about how we do it is to bring these different changes together so that we have a way of putting them all in together for each individual person. And I think that's quite a big challenge. So I'm just going to present some of the factors going forward. And surgery, it's needed for diagnosis at the moment. It's the gold standard. We do have scans and MRIs that help us, but at the moment, if we do surgery, it's unethical not to actually treat someone. So whenever we have someone with endometriosis, they have actually already had treatment before we can actually enroll them into any research studies or any studies about the pathophysiology of endometriosis. The next big problem is the control group, because we need to have had surgery to know that someone doesn't have endometriosis, but there has to be a reason they've had surgery. So quite often they have fibroids or other pathologies, an ovarian cyst or another pathology that's meant they've had to go to surgery. And we do use, in the control group, we do ask women who've had tubal ligations to be in the control group, but we don't do as many of those as we used to because of the Mirena coil. And so to have a surgically diagnosed control group is quite tricky. The other thing is that sometimes we use pain-free, healthy controls, but as we know, symptoms don't correlate to endometriosis, and some people with endometriosis really don't have symptoms. So unless someone has had surgery, we never really know. And one of the ways we overcome that is to have two control groups. One, a control group that's had surgery that may have some benign gynecological pathologies, and the other, a pain-free, healthy control group that hasn't had surgery. The good thing about surgery is it has helped us a lot with theories of endometriosis development. And Sampson, very early on in last century, noticed that a lot of women had retrograde menstrual fluid in the pelvis, and he theorized that tissue in this fluid stuck to the peritoneum, attached, and embedded itself and grew. And this is still the most predominant theory, There have been work in baboons which have modelled this theory, but it doesn't account for all endometriosis that is seen. And there's some very rare cases of men with endometriosis who've been on high-dose estrogen, and other theories about stem cells and coelomic metaplasia. So there are probably other ways of getting endometriosis, but predominantly we think Sampson's theory is the most common. But the big problem with Sampson's theory is that 90% of women will have retrograde menstruation, but only 10% develop endometriosis. So what we don't understand is why those 10% get endometriosis. And it is a really big challenge to try and work that out. And I'm going to try and show you some of the ways we have. First of all, one of the methods we can work out about the pathophysiology of endometriosis is to compare women with and without endometriosis and have a look at their biological samples and see if there are any differences. And when we do that, when we take samples of blood or endometrium or peritoneal fluid, we find there are very big differences. And that's in the cytokines. We see differences in epigenetic factors like microRNAs. We see some hormonal changes. So Serta Berlin has looked at aromatase activity in tissues, and it's quite different in endometriosis. We see immunological differences, and there are some factors related to genetic inheritance as well. And we can look at those differences to see if that can tell us why and how the lesions develop. When we look at genetic inheritance, endometriosis does have a genetic inheritance. It's more common in twins, and the early studies were done by Susan Traylor from Brisbane, and she looked at 215 twins and found that there was a 51% of the variance in endometriosis was related to genetics. And this was backed up by some very large geographical studies using entire populations in registries in some of the Scandinavian countries. And Stephenson published on 750 women with endometriosis who were more interrelated than in a matched control group. So there is some genetic inheritance. The difficulty is it's very hard to study. And one of the ways we've tried to study this is to use gene-wide association studies. There are 4 or 5 very large groups who have collected very large populations across the world. 4 of these populations are in Europe and Australia, and there's also a large Japanese study as well. And they have combined literally tens of thousands of patients' SNP polymorphisms to find out if people with endometriosis in that very large cohort have a higher SNP polymorphisms in certain genes that would contribute to the endometriosis process. After all the study, they have found, well, they last published on 12. They've actually found 2 more since this publication. And they found some polymorphisms in 10 independent genetic loci. However, these polymorphisms were actually in non-coding regions. And each one individually only explains a very, very small proportion of the disease risk variance. So it's a very polygenic study. There is some genetic inheritance, but it's not very easy to associate that inheritance with getting the disease. So there are other factors that have to be contributing to disease initiation and progression. One of the very interesting new thoughts in endometriosis is looking at somatic mutations. And Angelesio et al. In 2017 published in the New England Journal of Medicine, and Suda shortly after, showed that in the tissue, in retrograde in somatic lesions of endometriosis, some of the patients had clonal expansion of cells with a growth advantage. And these mutations were different in different lesions from the same patient. And 19 of the 24 patients had these somatic mutations. 5 of them were in somatic cancer-driving mutations, and these are quite proliferative. So these particular mutations drive growth and proliferation in the lesions. And interestingly, they were all confined to the epithelial compartment. So these mutations would confer a risk of endometriosis in someone who potentially didn't have a SNP polymorphism in their DNA. And this has been a really interesting area of study in endometriosis because it varies between lesions and it can be different mutations in the same person. We've also looked a lot at immune aberrations in the peritoneal fluid and in lesions from women with endo. And we find a lot of differences and a lot of changes in the immune system, but it's very variable between women. And it appears to change at different times in different places in different women. We see both upregulation of inflammatory immune cells and inflammatory cytokines, but also reparative profiles. And these tissues are very active. We see a lot of messenger RNAs when we do arrays and sequencing, and we also see a lot of microRNA changes, and much, much more compared to the eutopic endometrium. And these tissues are very active. We see cytotoxic as well as suppressive cytokines. So there's a lot of inflammation, but also healing and repair. And the variation is so high between patients that it's hard to get a clear picture. We can also look at the molecular biology, and we find we get a lot of RNA changes, but also microRNA changes. And we can use these changes to predict different pathways and targets, but we have to go back to the human tissues to figure this out. And these are all predictive. It's a structure for us, but again, it's something that we can get an idea of, but it's very hard to be really certain about this. And that's where we really have to go to animal models. So I'm going to spend a little bit of time talking about our animal models because they have really helped us move forward with exploring some of the targets for pharmacology in endometriosis, but also some of the ways we might be able to use to understand the disease process and how to treat it. There are several in vivo models of endometriosis, and probably the one that is most like human disease, and in fact, in baboons, when you take them out of captivity and prevent them from being conceiving, will naturally develop endometriosis, although it doesn't happen at a very high rate. Because of that, they do have an induced model of endometriosis in baboons. And the very seminal work by Tom de Hoog from the Netherlands really helped us a lot with understanding endometriosis in primates. Julie Hastings and Ezgi Fazialabis out of Chicago also had a baboon model, and their work has really helped us quite a lot in the most similar model to humans. The difficulty with working in baboons is they're expensive. You can't have very many animals, and there's some ethical issues around working with baboons. But one of the other things is you can't look at the lesions in a very short time frame. You have to perform a laparoscopy sort of 6 weeks to 2 months apart. So you can't really see changes in lesion development in a short time frame. Because of this, we've developed mouse models and rat models of endometriosis, and these models, none of them are perfect. We can use homogeneous models, and we tend to have to prime the endometrium of rodents and decidualize it for the lesions to take. And in some models, they have actually sutured the tissue or sutured little pieces of the uterus, which obviously is not the same as human disease. And the murine and rodent endometrium has a different structure to humans. So it doesn't really completely model the disease, but it gives us a rough a model of understanding, and we can keep the immune system intact in a homogeneous model because it's murine tissue and murine immune system, so you don't get rejection. In the heterogeneous models, this is where we use human endometrium, and we use mice with an immunocompromised immune system. The main cells that we don't want to have present are T cells and B cells. And in nude mice, there is no thymus and they don't make T cells, so you don't get rejection. In skid mice, they don't have B and T cells. So the immune system is not fully represented. However, the evidence in humans and in models is that it's mainly the innate immune system that determines whether an endometriotic lesion develops, and it's mainly the macrophages that have a high role in either rejecting the tissue or remodeling it. At the moment, there's actually a working party out of the World Congress of Endo, and we're all getting together to try and help people optimize the use of animal mouse models to help us in human disease. So there is some work going on at the moment to look at how to do this better and to optimize and really have better models. I'm going to explain a little bit how we can use these models to really take us forward with the pathophysiology. And one of the ways we can do that is to look at the longitudinal mouse model of endometriosis that we used to see what happened to lesions over about a 2-week time frame. We did use a nude mouse model and used human endometrial tissue. And we were able to look at the lesions histologically and using immunohistochemistry, but we also could look at the mRNA profiles and see what happened over time. One of the things we found was that there was a very consistent morphological pattern. And by day 7, you can see here that in the center of the lesions, there was a lot of necrosis, and the glands were breaking down and falling apart, and a lot of cellular debris. By day 14, however, we did have some cystic spaces which had become epithelialized around the necrotic tissue, and we got complex glandular epithelium and very much higher number of glands. So there was an inflammatory process and then a tissue repair process, and this was very consistent. We were able, because we had used both human and mouse tissue, we could identify the component that came from the peritoneum as being murine and the component that came from the endometrium as being human. And there are lots of histological markers and nuclear stains that can determine which lesion is which. Additionally, because we used Affymetrix microarrays, they have human orthologs and homologs. And so you can actually see when any transcript that matches to the human and murine match, like CD68 was present as an ortholog and homolog for human and mouse, you can actually determine which species and therefore which compartment these lesions came from. And what we found was we had very high levels of the murine ortholog, but very low levels of the human ortholog. And CD68 is a marker for macrophages. When we actually did an immunohistochemical stain for murine macrophages, we found they were around the periphery of the day 7 lesions, but by the time they got to day 14, they had spread out in a network in the central part of the lesions. And we felt that the way they localized, it very much looked like they were controlling the remodeling of the lesion. The other advantage of this model was we could actually identify the transcripts and see how they related to each other, and we could find transcripts that linked the human endometrial lesion with the murine peritoneum. And we felt these markers were really important because if we could disrupt the interaction between the peritoneum and the endometrium, we potentially could prevent a lesion developing. And so we felt these were markers that we really would want to target. And TGF-beta is a cytokine that very much promotes repair and healing by macrophages. And this was a key marker in one of our molecular networks. We were able to use the messenger RNA data to go to online ontologies to look at the function of all the different transcripts that were coming up at the different time points. And that really helped us work out a model of how lesions developed over time. And this has been developed by other groups as well and added to in terms of changing hormones in animals to see the lesions breaking down initially into a necrotic hypoxic lesion. You get a lot of neutrophils initially, but then macrophages coming in to clear this tissue. And then what happens is you get a healing and repair transcripts, and you see the macrophages behaving in quite a different way. We start seeing blood vessel development. We start seeing transcripts for fibrosis, but also for nerves. And there's a lot of neurogenesis as well. And then finally, we see the stable lesion again by about day 14. And others have shown that in the response to the cyclic hormones of the menstrual cycle, you get an increasing tissue breakdown. And the lesion goes around in a circle where the immune system's brought in every month to clear the tissue, and it becomes increasingly fibrotic. And you get aberrant vascular and neural growth. There has been another fantastic paper by Backy et al. In 2009, and she looked very much at macrophages. And again, this is where a mouse model enabled her to activate macrophages either classically, which is in a very inflammatory macrophage that will cause tissue damage, that will engulf bacteria and cause inflammation. And she could introduce these macrophages into mice to see how endometrial tissue would respond. But also she could actually then activate the macrophages in a different way where they were going to heal and repair, and they would be alternatively activated. And what she found was when the macrophages were very inflammatory, you got suppression of the lesions and clearance of the tissue. When they were reparative, the lesions grew and they became larger. And so the function of the macrophages was really quite important to the lesion development. And we have a specialized mouse that has a promoter in macrophages that glows green. and we can identify macrophages on the basis of their MAC green status, and we could co-stain the macrophages only and co-localize the markers for either inflammatory markers or healing and repair markers. And we did this in a MAC green mouse. And what we found was that the early marker of inflammation came up at day 4 and was lower at the other stages, whereas the markers for healing and repair were much more apparent later in the lesion development. And this very much fit our model of tissue breakdown followed by healing and repair. And you can see how we co-localized the green of the MAC green macrophages with the other markers that we were using. So we then went on to hypothesize that the main reason people develop endometriosis is that when you got retrograde menstruation, the inflammatory macrophages would go in and you would get an inflammatory response. But if they didn't efficiently clear the cells from the endometriosis lesion, you would end up then getting tissue repair, and the lesion would adhere and grow. And if the healing process went really well, you would get lesion establishment. This is important in fertility as well, because we think one of the factors is the cyclical process of tissue breakdown and inflammation followed by healing and repair means that there's an ongoing inflammatory environment in the pelvis, and that this makes it hard for eggs and sperm and embryos to function because they're having to deal with an environment that's quite inflammatory. We also felt that we should be able to manipulate parts of this process. And one of them was, if we could reduce the healing and repair part, we might find the lesions didn't heal as well and didn't get bigger. And the way we chose to do that was to use another knockout mouse. And again, this is something you can do in mouse models that you can't do in humans. And we chose a TGF-beta knockout mouse that didn't have a major cytokine in the healing and repair process. These mice are quite tricky little mice to look after, and they have a aberrant graft versus host disease if they have an intact immune system. So they have to be bred on an immunocompromised background. So the TGF-beta1 deficient mice have to have a SCID background, otherwise they don't actually live. So we had a colony of TGF-beta SCID mice that was in Adelaide that we could actually use to look at endometriosis, and we compared them to the homozygous wild-type mice for TGF-beta1 and also heterozygous mice. These animals have functional TGF-beta2, TGF-beta3, but also their macrophages are intact. And what we found was when we had homozygous TGF-beta1 wild-type mice, we had quite large lesions. In the heterozygotes, we had moderate-sized lesions, but the lesions in the TGF-beta1 deficient mice were very, very small. And this really, we also looked at macrophages, and we found that the macrophages were not being attracted to the lesions in the TGF-beta1 deficient mice, and that particularly the inflammatory markers were lower. So we concluded that host-derived TGF-beta1 deficiency suppressed endometriosis-like lesion development, and that there was a reduction in macrophage activity and an altered function. we decided to look at this hypothesis in a different way, because we decided to enhance M1 inflammation and try and make it much more inflammatory to see if we could suppress endometriosis lesion growth. And the way we did that was we looked at microRNAs that epigenetically moved the function of the macrophages towards an inflammatory profile. And the microRNAs that we knew were dysregulated in endometriosis, because we had done an array study to look at the differences. One of them was MER223. And this particular microRNA, when it was deficient, it promotes inflammatory activity. And so we looked to use a MER223 deficient model of endometriosis, which again, you can do in a mouse, you can't do in humans. We were fortunate enough that Erin Greaves, who we collaborate with, had a murine model, a homogeneous model of endometriosis. So we could use donors and recipients in mice that were either MER223 deficient or replete. To prime the endometrium, so it will take at an ectopic site with murine endometrium, you do have to prime it with estrogen. You then have to expose it to progesterone, decidualize it, and we use sesame oil to do this, and then remove the progesterone, and the endometrium does slough, and you are able to get the endometrium implanted in the mouse, and you get lesions. Normal unprimed murine endometrium will not take. And we looked at doing that in C57 wild type mice or MER223 deficient mice. What we found was in the MER mice, the lesion size was quite large at day 7. There was a lot of edema, and there were a lot of glands, and that as time went by, the lesion size dropped down. And in the MER223 mice, the glands became non-existent at day 21, whereas in the C57 mice, the lesions stayed a similar size, but the glandular development increased and increased. So there was quite a difference in both the lesion size and the glands. We also looked at the macrophages, and in the inflammatory model of MER223 deficient mice, we got a lot more macrophages at day 7, but by day 14, the numbers were much lower. And we did determine that immunohistochemically. So the model that we had for this MER223 deficient mouse was that at day 7, in the inflammatory environment of the MER223, there were a lot more macrophages that were of the inflammatory type. And the lesions were larger, more edematous, and there were a lot more macrophages in the stroma. By day 14, however, the macrophages had cleared and created a tissue clearance of the lesions. And there were still some inflammatory macrophages, but not very many of them, whereas the reparative macrophages had infiltrated the wild-type lesions and started to remodel them and develop the glands. By day 21, we had virtually no macrophages in the lesions in the MER223s, and the lesions were very small and there were no glands. Whereas by day 21, we had a moderate-sized lesion with a lot of glandular development. And we felt that it was both the increase in the inflammatory macrophages on day 7, but also the absence of the reparative macrophages by day 14 that had really kept that lesion in a suppressed state. We felt that the ability to clear the tissue was really important to not have a lesion develop, but also to reduce the healing and repair prevented the growth. So when you look at different factors that could lead to endometriosis in the context of this theory, there are a lot of things that we know are risk factors for endometriosis in humans that will increase the growth of an endometrial lesion. So if the endometrial tissue is highly proliferative, it may well outgrow the inflammatory immune system's ability to clear that lesion, and the tissue will stay and grow. And the factors that are involved in that are an estrogenic environment. Dioxin exposure increases aromatase activity. Somatic mutations with a growth advantage will do this. A high endometrial tissue load will increase the tissue. And also frequent periods will increase the tissue load and make it harder and harder for the inflammatory immune system to remove the lesions. We also know, and not many people realise this because they do use prednisolone in women with endometriosis sometimes, but Tom de Hoog found in a baboon model that when you give baboons with induced endometriosis prednisolone, you actually enhance the growth of endometriosis lesions. And this is very consistent with this model where the inflammatory immune cells will be suppressed by immune suppressors like prednisolone and glucocorticoids, and you'll get more endometriosis. But also other things could immune suppress, such as chemotherapy or cancers or viruses. And it may be some of these factors on some occasions reducing the inflammatory immune's response ability to get rid of lesions. The last thing is you could also have enhanced repair. And we did a study in mice looking at seminal plasma because it has a very high level of TGF-beta. And we found the lesions developed and grew when the endometrial tissue was exposed to seminal plasma and when the peritoneal environment was exposed to it as well. There are likely to be genetic factors that improve healing, and also factors that improve healing and the environment of the tissue may help. And we think that the combination of all these factors will determine if one lesion will develop and grow or whether it will be cleared. So increasingly, we're thinking that for different women, different combinations of factors could lead to different endometriosis lesions, and in some cycles you might get endometriosis develop and not in others, depending on all the genetic and environmental and immune factors that contribute together to decide whether a lesion will grow or not grow. So we think it's a lot more complex. And I think in the future, we're going to be really developing personalized care pathways to really try and establish what can be quite individualized factors that decide whether endometriosis grows or not. And it may be different for different people with different genetic backgrounds. There may be different somatic mutations, different environments and exposures in a particular month or cycle. And there may be other factors in terms of hormones and cytokines that change the way the immune system behaves. I think there are a lot of things that we are doing at the moment that will help us with that complexity. And there's some really interesting papers on training macrophages so that they behave in a different way. And we can train them to either be more inflammatory or less reparative in endometriosis. We are looking to subcategorize with phenome development, meaning that when we look at some of the genetic factors, there may be differences in the expression of the disease that we can understand and help us actually individualize that care. There is quite a lot of work on exosome delivery systems where we deliver epigenetic factors like microRNAs and factors that will modulate the immune system or modulate the endometrial tissue so that it's less likely to become a lesion. We do have a lot of new pharmaceuticals coming out, and we have the GnRH antagonists that are becoming increasingly available for endometriosis. And we've been looking at some new pain modulators that are not hormonal to look at modulating pain. There's some very exciting work in cancer looking at vaccination for somatic mutations, and they may be applicable to endometriosis. And finally, we're using highly complex artificial intelligence systems to be able to put all this complex data together in a very personalised way for people. And that we're working in that area at the moment, and it's been very, very exciting. These are a lot of people who've worked with us in the lab with helping us obtain tissue, working on microRNAs. Kavita Paneer is currently a postdoc with Erin Greaves, who's done a lot of the mouse model work, as did Zahid Mohammed, and Maria Olsen-Teague and Wang Zhao did a lot of the microRNA work. And we have some really great collaborations with Chris Print, who's a professor in Auckland, and Steve Charnock-Jones at Cambridge. Just to talk a little bit about what we're doing at the moment. We've had some very exciting time, and in fact a large grant last week to look at better diagnosis so that we don't need surgery as the first step in determining whether someone has endo or not. And we did a very large Cochrane review looking at all the different markers. There were 15,000 papers, and specialist endometriosis ultrasounds and specialist MRIs had the most sensitive and specific diagnostic capability, but they still weren't good enough to replace surgery. And we met with Gustavo Carniero, who's a professor in the Australian Institute of Machine Learning in Adelaide, who was very interested in putting the digital data for both these modalities together to try and improve diagnosis non-invasively so we can understand lesions a lot better and how human disease changes. I've got some amazing collaborators here. Jodie Avery and Beck O'Hara have done a lot of the work getting the scans and organising our collaborations. We've got amazing people who've helped us fund our grants. We work with the Jean Howes Institute, which is a national NGO and has a registry for endometriosis patients, and we're hoping that they will upload their images and provide informed consent that we could potentially use them in our AI models. And then we've got some amazing imaging experts, both in Adelaide, and the three on the bottom here are experts in ultrasound and MRI. And George Condis is in Sydney, and Matthew Leonardi is in Canada. And they're really helping with us with this project. One factor in putting this project together is that we had very much a lot of involvement from all the consumer groups in Australia, who luckily lobbied the members of Parliament and indeed the Prime Minister, whose wife has endometriosis, to help with funding and help really move forward with our understanding of endo, but also being able to diagnose it without surgery. But also looking at some of the new developments that might actually be therapeutic for people as well. So I have to thank all of these people. That's the end of my slide. I shall end the show. And I do hope I've explained everything.
So this is, thank you very much. This is one of the great lectures we've just gone through listening to so much science behind this enigmatic epiphenomena, benign cancer disease which we have. And I hope we can, with all this research, find a cure very soon with it. And the most exciting part, which I heard, was the pain modulators, the vaccination, the GnRH antagonist, and of course, your artificial intelligence. We have a lot of questions, and I would request Dr. Pratap Kumar to kindly put those questions to Dr.
Thank you
Halvi.
very much for your wonderful lecture, Professor Mary Vishal. You stressed upon macrophages and inflammatory cytokines in your talk, and you also did mention about microRNA and the recent research. So the first question to you is that when we know microRNA can be an invasion or it may be suppressing, if you look at the number, you said about microRNA 223 deficiency causes endometriosis. What about the other microRNAs? MicroRNA 145. Do you think 145 has a place in the future?
The microRNAs are really interesting because they really fine-tune the translation of messenger RNA to protein. And I think, and they are transported around our bodies. So cells will secrete them, and a lot of the immune cells will actually use them to signal locally to cells, but they will also release microRNAs into the blood system. And I think a lot of the systemic effects of endometriosis are probably modulated by markers like this. And they're secreted in little packets called exosomes. So I do think microRNA 145 is likely to be involved, and we've certainly found it dysregulated. The question is to work out how and can we use them, and the way we use hormonal manipulation, we use the body's system by introducing hormones. Can we introduce these microRNAs in a way that targets the right cells and is specific to endo? And they're looking at some of the markers on the surface of the exosomes and how our bodies deliver them to certain cells specifically. And I think once we learn more about that, we may well be able to deliver some molecular tools to the right cells. But one of the big problems with endo is having a specific delivery system, and we haven't developed that yet.
Thank you very much. I like the way you said the little packets of exosomes, and we might be putting microRNAs into those packets and send it to the body. I think that's an advance, I don't know when you're going to see that. Interestingly, do you think the same microRNA seems to be the big hot topic? Do you think it can be used for diagnostic and analysis of the endometriotic situation in the future?
We actually looked at plasma microRNAs, and they've probably been, our group published in JCEM, and then Tom de Hoog's group, a guy called Arnie van High, published reasonably shortly after in Human Reproduction. But Hugh Taylor has also looked at a spin-off company. It's interesting because Arnie and our group, our data is very similar, and we did find differences in the expression, but they weren't sufficient for a diagnostic test. There was too much variability between patients. Hugh Taylor's group is looking much more commercially at that, and certainly has claimed to have better markers that are more effective, but we couldn't find that in our data sets. So I actually went to Belgium literally before I came to India, and Hyderabad, days before, to mark Arnie van Hig's PhD, and the data we found our data was fairly similar. He has done a review in Human Reproduction, looking at the reproducibility of the plasma microRNAs, but we were not able to get something that was diagnostic.
I think so. Another interesting topic you discussed was how we need to probably in the future train macrophages. That's very interesting. I think it's all connected to exosomes, and somehow we need to train macrophages, according to you. So, how do you think it will happen?
There have been a couple of papers that have published about exposing macrophages repetitively to different stimuli to get them to functionally behave and then introduce them to be more inflammatory or more healing. And they've got different methods of doing that, but there's been 2 or 3 papers in the last 2 or 3 years looking at, manipulating macrophage function through exposure mainly to things like LPS that are really interesting. And I think that's something that is really an interesting thought, certainly in endo.
Yeah, Naren, you have some questions.
So if we can train these macrophages against endometriosis, we could probably try and heal many other diseases by like this. Especially, do you think something like this could come for COVID treatment or antiviral also train them against attacking specific viruses and things like that in the future?
Yes,
malignancies.
it's a little bit. I mean, it's a little bit hard. I don't know that I can comment on. I would hate to think I would be an expert on COVID, but that's way outside my area of work. But it may be that we can teach them to be less inflammatory. Like I think some of the complications of COVID are a response to over-inflammation, and I think some of, not the vaccines, but the other medications they use is to suppress the inflammation to some extent so they don't cause so much tissue damage. I'm just not sure, but yeah, it would be interesting, certainly for endo, if we could, at the right time when there was tissue sitting around, we could clear it better and have a bit more sort of inflammatory clearance and not heal it. If we can change that somehow in a medical way, and we haven't got the tools, but I'd love to think in 10 years' time the technology develops in a way that we could try and target that. And if we could specifically get the macrophages to go to the endometrium and to the ectopic endometrium, that would be great.
Yeah. That's interesting. You did mention about aromatase inhibitor, and do you think the intracrinology needs to be understood a little better?
Yeah.
I think the new concept of intracrinology, and we've only heard about endocrinology. So what about intracrinology and aromatase inhibitors?
Exactly. It's very interesting. Thank you, Marie-Louise. Very, very interesting during the research. And for endocrinology, it's new future for the progress in this disease.
I agree. I think certainly Berlin's work very much suggests in the tissues aromatase is upregulated. And aromatase is actually upregulated by by COX-2, which is an inflammatory marker as well. And we think that might be partly why the endometrium's resistant to progesterone. Because there's more the estrogen progesterone balances out a little bit, and why implantation can be affected a little bit. But yeah, I think there's some really interesting work in that area as well. And Bruce Lessey often uses aromatase inhibitors in the follicular phase and additional supplementary progesterone in the luteal phase for people with endo. So that's where some of the progesterone-resistant work has come out, which is kind of interesting too.
So do you think the reason for progesterone resistance in endometriosis is basically connected to microRNA and its problems?
I think it's more upregulation of aromatase and inflammation causing estrogenic changes, more estrogen around, and then you don't prime as well for the progesterone. The progesterone doesn't have the same effect as quickly. It would be really interesting. I did ask some people from EV if, the phase shifts in the ERA test when you're out of sync, if it happens more in endometriosis, and they really didn't think that was true. But I've had some people with endometriosis where the ERA test is a wee bit shifted, and I just wonder if some of that is related to progesterone resistance.
Yeah, because I'm finding in clinical practice when an endometriosis doesn't respond to the response, I just try letrozole and it works actually. That's maybe the reason inhibitor goes down and the pain goes away.
I think that's probably true. And certainly Bruce Lessey, who kind of developed the progesterone resistance narrative, he uses aromatase inhibitors quite a lot for that.
What about people are thinking it's all connected to newborn bleeding for a female baby. Do you think these kind of an endometriosis is seen because of these kind of cells which goes retrograde in the newborn itself?
I think that is Caroline Gargett out of Melbourne, and one of the implantation guys out of London have developed the idea that the stem cells in newborns are very, very potent. And when you get the estrogen and progesterone withdrawal and some bleeding at delivery, those cells may be retrogradely going into the peritoneal cavity and seeding it at that point. I guess the things that support that would be the fact that 38% of women with endo have pain from their first period, and something like 60% are under 20 when they get pain. So that goes with something happening pre-puberty. I'm just not sure what the evidence is there, but I do know Caroline Gargett's work very much suggests endometrial cells from very young people, like newborns, are highly proliferative and pluripotent, and that could be seeded. So it's hard to know for sure, but a lot of women have problems right from their first period. Their life has been easy. They get their first period and life changes. So it would very much go with the narratives we hear from the consumer groups.
Yeah, true. Narendra Malhotra, do you have any questions?
So this is something which has been excellently brought about, and especially the newborn stem cell theory and also the progesterone. We thought that giving progesterone would be, but the PRAB and the B receptors theories, there's so many great things in endometriosis which are still to be understood. But I'm very excited about the artificial intelligence being brought in and the various customized treatment which might come in, like with pain modulation and of course antagonists. So I thank you very much for this. Would Yvonne like to conclude this, and Jaideep?
It was wonderful listening to you and discussing. Yeah.
Thank you.
Fantastic.
You very much.
Really fantastic.
Excellent. Great.
Thank you so much for inviting me. It's been a real pleasure. It's lovely to catch up too. It's just a pity we can't meet in person.
Yeah.
I know, especially when Australia is locked to all of us. I would take the first flight and come to Australia and New Zealand.
I left Hyderabad at the Indian Society of Reproduction conference literally 4 days before Australia got locked down. And no one could go anywhere.
I know. You just made it back.
I nearly didn't.
Yes.
But I had a lovely conference there, and I'm so glad I had the chance to do that right then, because it's so much harder not to be able to. But it's amazing.
Jaideep and Yvonne, can you conclude the session? Jaideep and Yvonne, can you conclude?
Yeah, I just wanted to ask, Louis, I want to ask you one thing. All of us do such a lot of work. How is it that with all the clinical work, also you all are able to do so much research? Throw some light so that our young people can understand, the value of research and taking it forward from here.
Probably my research and my clinical work integrate very well. So a lot of people I recruit through my clinical practice and I help people with endo in my IVF practice. It integrates very easily. I have got incredible. I've just spent a lot of my life being able to be surrounded by incredible nursing staff, great researchers, and I do actually have a really good husband that feeds me properly and makes sure I go to the gym and things like that as well. So you just have to. I'm very lucky.
Here we both are feeding each other, so we have both no time to no time for it.
Then you have to train your children well.
No, but from the, I'm asking from the organization front, like, a lot of grant must be coming, and then, directed research on, so how does it all work about?
So the Australian government has actually changed a lot how we do grants. So essentially, there's a lot more focus on collaborative groups to be funded. So it's almost impossible to get a grant without involving the consumer groups. And Beck O'Hara actually runs a large forum, but she's integrated. She works very closely. She does a lot of qualitative research and lobbying with the consumer groups across the country. And we're building a website to represent Australian. It's a hub for all the different consumer groups, but also the researchers and the clinicians. So we've networked with that because we want them all to integrate on that platform. The Nest registry is a new initiative that's been funded by the government as well. And Jason Abbott and Cecilia Ning run that. But what they're wanting to do is for clinicians and women with endo to put a lot of data about life course events. And they're hoping to use big epidemiological methods and AI to look at, when you get lots and lots of people, you can say, well, when these people had a marina, they did better with this than those people who didn't. But you need huge numbers. So they're trying to set that up. The same with the imaging stuff. We need very large numbers of scans and MRIs for the imaging guys to pick up patterns in the data sets. And randomized controlled trials are almost impossible for endo because everyone has a different phenotype and different symptoms, and they're all treated already with surgery. So it's very hard to do them, and that's where these kind of big epidemiological methods are required. We've been very lucky. We have a cross-party group of women who are federal MPs who lobby for endometriosis, so they have quite a lot of ability to help us politically. And then we've been actually quite lucky because our Prime Minister has an experience of endometriosis that has meant it has helped us a lot. But we haven't been able to talk about endometriosis very often. And the consumer groups feel with social media and the ability to talk about menstrual health and periods and all these taboo topics that women have found really hard to talk about. Pain and menstruation are not easy things, but we've had some very amazing advocates in the community who are prepared to tell their stories and say how they couldn't get to school. And we've also now got a schools program for endometriosis and menstrual health that my ex-PhD is running. And that means that boys and girls, when they're young, have a language to talk about endometriosis. And when they're older and their employers and their colleagues have endo, they can talk about it, they can understand it. And we've made sure that men and women get the information when they're young. And it's quite different.
Okay. Yvonne, you have any questions to ask or any comments to make?
No, only thank you very much, Mary Louise, because you shared
Okay.
very interesting in intracraniology. And for FIGO, the future with this research is very, very interesting too. Thank you very much.
Okay. I have a lingering question in my mind, Lois. You know, what about improving the egg quality or results in IVF? Is there what is the research moving on towards in this area?
There is quite a lot of information where they have used animal oocytes, or some human actually, where they have put peritoneal fluid around the egg quality, with the eggs, and it's quite clear that there are mitotic spindle changes and changes in that inflammatory environment. And we always think with endometriosis it's a delay to conception because of the environment. So changing the environment is a way of improving. That's where I think surgery helps, because you get rid of the lesions, you get rid of the inflammatory triggers. IVF helps because you take the eggs out of the environment. And there's increasingly some work looking at something called lepiodol, or there's a tubal flush with some poppy seed oil that people feel probably has an anti-inflammatory effect, and it lingers in the pelvis for a long time. It's got a very long half-life.
Yeah.
And the Dutch found it improved fertility rates and unexplained fertility, but we know that unexplained fertility, about 80% have undiagnosed endo. So there's a little bit of work thinking that maybe there's some medical treatments that might help people with endo.
And what about progesterone resistance, which is there? Like, it's an emerging, not really an emerging, but it is a concept. So why does that happen? And can something be done?
So the Bruce Lessey, who is a lovely guy, he works in America. He's sort of, and Sir de Berlin are kind of involved in the progesterone resistance changes. I think the systemic effect of inflammation upregulates the inflammatory pathways, the NF-kappa B pathway. So you get high levels of COX-2, which upregulates aromatase. So you get more estrogen, and the ratio of estrogen to progesterone goes out a bit. So women often find they get premenstrual spotting. Yeah. And that will settle down with progesterone. So I tend to supplement in the luteal phase to try and help that. Bruce Lessey also talks, like Dr. Pratap was saying, he would advocate using aromatase in the follicular phase to suppress estrogen a bit and get that balance better. And I do think sometimes that really helps when you are not looking at IVF, when you look at more conservative management, to be able to track a cycle. Obviously, letrozole will also give you possibly another egg if you're not careful. So there's an advantage there as well. But he feels the advantages for the endometrium. So
yeah. You just have another question from the audience. Can I ask that one or two questions? Do you think that the iron sequestrated by the macrophages as ferritin is a pro-oxidant, and thus the iron-inducing oxidative stress is seen? So do you think you can blame once again, the macrophages and the ferritin kind of a reaction?
There's certainly a lot of evidence that iron is increased in the lesions. I think what happens also is one of the reasons you get red and black lesions, that as that necrotic central core breaks down, sometimes there are blood vessels that will bleed. And so I think some of the iron probably comes from hemoglobin and heme. And that's where you get, in the lesions, you get hematoxylin, and you often find it in the macrophages. So the macrophages tend to try and clear it. I do think it's all part of the inflammatory area, and you get oxidative stress from that that dead tissue, but also iron in that tissue. So that's how I think of it. But
I think so.
Yeah, other people
And
think differently.
there is one last question, seems to be a little difficult. There is a considerable interest in the development of an inhibitor of aldoketoreductase, AKR163, IC3. Maybe the pharmaceutical people like Subhojit may be interested to know the future of inhibitor of aldoketoreductase.
I know that there are a lot of modulators of pain. So we've had toll-like receptor inhibitors and things like that. I'm not that familiar with aldoketoreductase, I'm sorry. But I would love, if any of you know, I would love to know what that is. So I think there's some really exciting pharmacological developments.
Yeah, it's one of the research which is going on, I think. So we'll have to wait for the results now. Yeah.
I see.
I
think that brings us to the end of the session, Pratap.
Yes, we're finished.
Yeah, so thank you, Louis. That was really an amazing session with great interactions, and I have to thank you from the bottom of my heart for a wonderful, wonderful session. And thanks for being there. And thank you to Pratap, Yvonne, and Naren for always being there for our this masterclass, and FIGO especially. Heartfelt thanks to the FIGO executive for being able to do all this. Thank you so much. And thank you, Shubhajit and Acumentis, for all the support, and DocMod also for all the support in this event organization.
So thank you all. Bye-bye.
Thank you very much.
Thank you. It's been a real pleasure.
Thank you.
All the best. See you soon.
Thank you.